Hello students, welcome to Civil Services Gurukul, a new initiative which is garnering support from students like you. And today we are back with, as promised, a new current affair update. So yesterday we had discussed the historic homecoming of Sunita Williams, but today's topic is from biotechnology section. We all know in last 4 or 5 years after space, it is the biotech section which is dominating UPSC civil service examination. And we all know prelims is just around the corner, so you cannot afford to miss any current updates related to biotechnology. So without wasting any time, let's start today's biotech breakthrough, which is making headlines for all the right reasons. So we'll be discussing genetically engineered woolly mice. So as you can see in this particular image, these are normal mice only if you look at the size or the basic, you could say the anatomical framework. But if you look closely, it has wool on its surface, unlike the conventional mice. So mice has been a common animal from times immemorial through which lab or you could say scientific experiments have been carried out. We had discussed that in class as well, that why mice is chosen over a lion, tiger, elephant or any other species, because it is easy to manage. There is no restriction regarding if anything goes wrong in experiment, because this is one of the species which typically not listed in Wildlife Protection Act, Killings Act, or you could say in the schedules. And it has fast multiplication rate, so you can observe future progeny if there are any mutations or anything which goes wrong, which is reflected or not. And also it is economical to maintain, and most important thing, it's a mammal which shares close, you could say genetic and protein framework with all other humans as well. So mice has always been a go-to choice for experiments. Now, why this experiment has made headlines? Because in this case, the first word, the woolly, is thing which has not been created ever before. Now, if you look back in animal history, the species which will come to your mind with the starting with prefix woolly is the great woolly mammoth. So this breakthrough was performed in USA. To be specific, it is the Colossal Biosciences in Texas which has performed this groundbreaking experiment in which they have used the advanced tools of, you could say, gene editing to perform this specific experiment. So as you could see, that woolly mice was performed using a CRISPR gene editing technology, which we have seen in last 5 years that this technology is making headlines because it is precise, it is economical, easy to carry out, and it has made genetic engineering a cakewalk for all top researchers. So overall, we can say that this CRISPR-Cas9 has already received Nobel Prize few years back. They were, would say, French scientists, Carpentier and Desmodona, who had initiated this breakthrough. But today, this technology is finding its use in agriculture, medical sciences, industry, and all other plethora of experiments. So in this case, they use this specific gene editing technique to insert gene which was frozen from woolly mammoth into the particular mice, and they tried to mimic its woolly fur interface. So almost we can say the human beings are acting like God, which raises some ethical concerns nevertheless, but still its breakthrough is quite significant, as we will discuss in the next slide. First, let's go back. What exactly was woolly mammoth? As you must have seen in Ice Age or a lot of animations, it is a typical, you could say, large size elephant which was even larger than the conventional largest terrestrial animal, African elephant. And what made it distinct was its woolly fur and its, you could say, specific tusks. So tusks of elephant, we all know, are modified incisors, and in this case you can see they were quite specialized and they were quite curved as well. So this species became extinct. There was a time in middle Pleistocene when it was dominating the Earth landscape, but coming with the Holocene epoch, it became extinct. So in 2015, there is a landmark project which is trying to revive woolly mammoth once again. A lot of environmentalists and scientists feel that this is not the right way to go about a species which has already become extinct. Why are we trying to revive it? Because more or less it will not likely to adjust in current climate change scenario, and also to bring back something that has been extinct is frowned upon because they feel that it is playing with nature as well. So in this particular experiment, it is not that this experiment in a way will bring the woolly mammoth immediately back, but it is a stepping stone, because what they tried to do in this particular experiment was they tried to isolate a particular gene which is responsible for this specific fur which comes to the elephant, and they have mimicked it with perfection. You can see the fur is being replicated on the mice body, which typically no other mice has as such. So even though the techniques are not to be memorized in GS perspective, but still to develop an understanding, I am giving you a brief overview how this landmark experiment was carried out. So as you could see, the first of all, we all know the hereditary information of any specific species, it's present in their DNA, which we call deoxyribonucleic acid, which is present in the nucleus of any living cell of that body. So what they tried to do was, because we know there are frozen samples available of woolly mammoth and they have stored that, so they isolated that particular DNA segment from that segment, and at same time use modern Asian elephant DNA segment as well, because current genome sequencing technology has made it possible to decode things. You all have heard about the epic human genome project, in which we deciphered that we have almost 20,000 genes in our body. So in this case, they just compared the DNA architecture. We all know that nitrogen basis sequence ATCG we typically refer, and then when they compared, they finally found out that which are the specific genes which are not there in Asian elephant, which are there in woolly elephant. So out of that, so they were able to decode that these specific genes are more or less likely to be responsible for the fur structure, because in cold, as a cold adaptation, you need to have a fur so that the heat loss is prevented. So finally, I cut those specific segments of gene and then use CRISPR-Cas9 technology to insert that in a specific mice, which was a normal mice, which we use in various lab experiments. So, but the real test is, it's easy to insert, cut and paste DNA segment with these gene editing tools, but the real success is whether it will be able to emulate that specific trait which we are trying to encounter. And with this, these scientists at US has achieved that breakthrough, and this particular mice you can see is transformed. So any species which carries foreign gene is typically referred as genetically engineered or RDT modified species. So it is a kind of a transgenic animal, because it's carrying all the genes of mice, but it is carrying the few segments of woolly mammoth which have been able to reflect on its architecture. So you can say this is a quite a new breakthrough, because we have seen BT cotton, you could say GM mustard. These are all plant experiments which are carried out, but this one is a quite unique in its own endeavor. And coming to the main part, because as a UPSC aspirant, you should be able to develop a holistic view that this particular breakthrough on a rat, what is larger significance? Should we morally support this development, or this is a slippery slope in which we are leading to some designer baby angle, or you could say it's a ethical doom which we should ideally avoid? So scientists have defended this move with four specific reasons, and these are from mains perspective. For pre-point of view, this topic is just going to be asked what exactly is a woolly mammoth, why did it become extinct, or what are the significance of rat. From mains perspective, gene editing as a protocol or CRISPR-Cas9, you could say theme, it can be asked that what is the scope or potential of this technology to make our life better. So first thing that they have highlighted from this experiment is that it's a given experimental proof that concept of de-extinction is possible. De-extinction is a modern day term in which we are trying to revive a species which has become extinct. We all know even in India in 1947, due to excessive hunting, cheetah got extinct, and today we have reintroduced African cheetah, though a lot of environmentalists feel that it's a invasive species which have been introduced. But through this technology, we can revive the extinct species if we have a selected sample of DNA from any of their body part. It has been preserved meticulously. So basically, that specific genetic traits which we only assume that existed can be replicated in a real life animal. So because we all know the diversity is quite unique. Already there are over, you could say, 2 million species which are in fray, and each species has some unique function which it performs, and through these experiments we can replicate those. So This opens a Pandora box of large options which can be explored to produce some new species. So the first basis of de-extinction, it could be a new theme in itself. The second reason why this experiment will remain significant in coming time is because it showcases the next step of genetic engineering. So we every decade we can say is remembered for some breakthrough. So who knows that this could be a new technology which becomes suddenly next go-to technology for genetic experiment. So this is another complex genetic interaction, and we can use this template for physical adaptations. And we all know the current climate change crisis is genuine one, and lot of species, especially these lower one, will not able to migrate or adapt to the new normal of temperature. As you can see, it's just the last week of March and we are witnessing almost May June type climatic conditions. So in case of animals, they can be used so that they can better adapt to the new normal of the climate change. So basically, this technology can be used constructively. It's not that they are just trying to mimic some odd species in some lower animal without its consent and trying to just replicate. So it could be used to save such species who will not be able to migrate or adapt to new normal. So that's the real breakthrough significance of this discovery. The third potential is that it technology can be used for biodiversity conservation. We all know biodiversity conservation mainly operates at two levels: either it's in situ or it's ex situ. In situ, even though has a better success rate because in that species evolved itself, and we can say national park, biosphere reserve, wildlife sanctuary have been go-to technologies for conservation. But still, in extreme situations, when we feel that predator risk is very high, poaching risk is very high, or we feel that we are not able to produce what exactly is required, then there is no harm in relying on ex situ technologies, right from the zoos, botanical garden, or you could say the cryopreservation. So this realm can be included in that domain, in which through selected species we can carry out these experiments so that their journey on earth is prolonged, because currently the sixth mass extinction is underway and it is mainly due to human reason. That's why we call it anthropogenic extinction. So this technique can be used to prevent extinctions, and we can just supplement these species effort for adaptations. And the final one is, this could be an ideal model for studying cold adaptation, because in this breakthrough we were finally able to just specify exact genetic segment which was responsible for producing fur in that woolly mammoth. So finally science is able to decode exact genes, which one is responsible for height, which one is for eye color, which is for skin tone. And once you are mapped the specific gene, and if we are able to successfully incorporate in another species, we can produce some marvelous examples, and they could be our next level solutions for plant as well as animal biotechnology, because currently the scope is immense. We can produce insulin, hormones, enzymes on mass scale by isolating specific genes and producing them in this animal, because cloning and other technologies at times are quite, you could say, rigorous and costly, but this method can be replicated for larger purpose as well. So we can say that genes, how genes are influencing thermoregulation, it could benefit wildlife conservation in extreme climates. So overall, we can say that this experiment has made science open new frontiers for you could say endeavors for socio-economic development. Even though it's an eye-catchy one, this will almost be something like the Dolly the sheep movement for biotechnology. We all know the Dolly the sheep was first cloned animal which opened new, you could say, dimensions of cloning, and after that there were plethora of animals, right from dog, Afghan hound Snuppy, to the CC copycat, to the Ralph the rat, and to Tetra the monkey. Even though in India also we are being successfully carrying out cloning, so hopefully Indian scientists might also replicate this model in some new species. But rat and mice continue to dominate the experiments, and once again this rat has garnered all the support of scientists who are progressively supporting biotechnology, even though little controversial, because you can say no consent is taken and we don't know the future how this rat will evolve. But at the moment we can support this experiment for larger purpose. So I hope this update on biotechnology was a useful one in some way, and make sure you just don't listen, update them in your class notes as well. And also this specific PPT has some, you could say, significant part. So we are starting a Telegram channel on Civil Service Gurukul as well. The link would be shared in your description channel. So those who are interested in joining are most welcome, because we will be uploading the current affairs and their PDF part in their Telegram channel as well, so that you could benefit and access as per your needs. So we'll be back soon with another sort of videos. I hope you keep viewing as per your convenience. It's our humble endeavor to support students, and hopefully we'll stay connected for more. So make sure you also subscribe our channel so that you get regular updates. Thank you very much. Have a nice day.